Hello, students. This is our lecture on stem cells, section 10.4 of the textbook. And really, what are stem cells, what are the different kinds of stem cells, and what are their potential uses? You're going to need a full piece of paper and obviously a pen or pencil to take down the notes. There's not a whole lot of note taking. And we're going to do that intermittently as we watch a series of video clips. I know it's a little weird to watch a video of video clips, but that's it's a convenient way of addressing the basics that we need to know of what stem cells are. So if you need to pause, please do so. Grab a piece of paper, have your pen or pencil ready. Most of the video clips are pretty short, about a minute or two long. The first one is the longest one, just 4 minutes long, and Hank Green is going to explain to us and introduce the concept of what stem cells are.
Okay, so you're reading the newspaper, you're watching the news, and they're talking about some new medical technology, some breakthrough treating congestive heart failure, regrowing muscle tissues in wounded soldiers. I bet you that story is going to mention that this new type of therapy uses stem cells. And I bet you, like most people, are going to listen along and be like, yeah, oh, yeah, oh, yeah. Without actually knowing what stem cells are, because who has time to know what stem cells are? Today, we're making time. You got lots of different types of cells in your body. You got muscle cells and skin cells and liver cells and brain cells, and most of these cells have to be replaced every once in a while. Your taste buds, for instance, are replaced every 10 days or so. Skin cells are replaced every couple of weeks, and liver cells turn over every 300 to 500 days. The cells that are doing the replacing of the old cells and the repairing of the damaged tissues are adult stem cells, also called somatic stem cells. The different sorts of cells, skin cells, liver cells, retina cells, muscle cells, intestine cells, they all have very specific jobs, and they're built in very specific ways to do those jobs. Different shapes, sizes, contents. You can't just stick a muscle cell into a damaged liver and expect it to start breaking down your alcohol for you. Somatic stem cells, on the other hand, haven't decided what the heck they're going to be. They're undifferentiated. They haven't specialized yet, like a college freshman. Or, let's face it, a recent college graduate. They have no idea what they're going to do with their lives. So just like there are different types of college graduates, there are different types of adult stem cells. Some can become more different kinds of things than others. Pluripotent.
All right, let's pause it there for just a second, and then let's go to our first note. All right, so here's our first note. So title your notes, section 10.4, stem cell notes. And then do write that question down at the top. What are stem cells and how can they be useful? So those are really the two questions we want to address in these new notes. What are they? How can they be useful? There's our definition of what a stem cell is. So these are cells, they're unspecialized. And they so unspecialized cells from which differentiated cells can develop. Excuse me. Well, what does differentiation mean? Differentiation is just the process by which cells become specialized. All right, so what does this mean? Well, take, for instance, your skin cells, right? Hank talks fast, and he mentioned a bunch of different kinds of cells. Let's just focus on skin cells. When you cut yourself, those skin cells obviously need to heal, and then they grow and divide until that cut is healed. Even just every normal day use, cells wear out, and they need to be replaced. But a skin cell is specialized, meaning that it is only going to divide into a skin cell. One skin cell will divide into two skin cells. A skin cell isn't going to divide into a muscle cell or a bone cell or a liver cell. Same thing with bone cells. Bone cells, when they divide and grow, are only going to divide and grow into bone cells. So they are specialized. Stem cells are cells that haven't become specialized yet. A stem cell can become any other kind of cell. Hence the term stem, like it's at the root of a stem, and that stem would branch. It could branch into that stem, could branch into many different branches. That cell could become many different kinds of cells. So just our first note, a stem cell is a undifferentiated cells from which differentiated cells develop. So skin cells develop from stem cells. Bone cells develop from stem cells. Liver cells develop from stem cells. Once they are specialized, that cell can only give rise to that kind of cell. All right, let's keep going with Hank.
Stem cells can become many different types of cells all over the body. However, they're really hard to track down because there are so few of them in each organ or tissue. There are also multipotent adult stem cells, which are more common in the body, but restricted in the kind of cell that they can become. It's kind of like the difference between graduating from a trade school, where you've been trained to do a few different possible jobs, and graduating with a degree in philosophy or something. Equally unprepared for all jobs. So yeah, stick a pluripotent cell in a damaged liver, and it just happily becomes a liver cell. Pretty cool. But there are some even better types of stem cells to be had. Embryonic stem cells, which are also pluripotent. These are the cells inside a human embryo when it's a blastocyst. Basically just a tiny nugget of human cells for...
All right, I'm going to stop this here for just a second.
5 days.
Nothing to write here. I just want to explain what a blastocyst is. So sperm fertilizes egg. And then after about 4 or 5 days, that one cell, sperm fertilizes egg, that one cell divides into 2, those 2 divide into 4, and so on and so on and so on. When it gets to 16 cells, it starts to form this sphere of cells or ball of cells called a blastocyst. So we're cutting through the blastocyst. This is about 200 to 300 cells in size, 4 to 5 days old. And just explaining what this, we don't need to know any of these terms, but so these cells on the outside, this is what would actually develop into the supporting tissues and the placenta that would keep the baby alive. And then these cells in here, this is called the inner cell mass. This is abbreviated ICM. These are the cells that actually become the baby. So these cells become the placenta and the umbilical cord. These cells actually is what becomes the baby. That's just a blastocyst. At 4 or 5 days old, we were all one of these things. The most useful stem cells come from the whole thing, and then embryonic stem cells come from these. So embryonic stem cells come from what we call the inner cell mass.
Old, which is destroyed in the process of removing the stem cells from inside it. These embryonic cells are obtained from in vitro fertilization clinics that fertilize eggs outside of a mother's body for couples who are having trouble conceiving. Naturally, these clinics have some leftover fertilized eggs, so with the donor's permission, they're given to scientists doing stem cell research. Now, the main advantage of the embryonic stem cells is that while adult stem cells can be grown in culture for a time, meaning they can be made to multiply over and over in a nutrient solution, they can't grow for as long or as fast as the embryonic stem cells, which can be maintained indefinitely under the right conditions. After just 6 months in culture, a single wad of 30 embryonic stem cells will have yielded millions of stem cells, which can go on to develop pretty much into any type of cell in the body. Also, adult stem cells, if used in some sorts of transplant therapies, are more likely to be rejected than embryonic stem cells. Stem cell research is currently pretty hoppin'. Embryonic stem cells are being used by researchers all over the world to figure out how to repair or replace damaged cells and organs and create new drugs, but regulations have taken their toll. There are only about 35 stem cell lines or families of identical pluripotent stem cells that are available for federally funded research in America, whereas Europe has a couple thousand. So there, now you'll never have to your way through another news report about stem cells again. Thanks for watching this episode of SciShow. If you have any questions, comments, or suggestions, you can find us on Facebook and Twitter, or of course down in the comments below. And if you want to keep getting smarter with us here at SciShow, you can go to youtube.com/SciShow and subscribe.
All right, so we made it through our first one. That's our longest video. Let's turn our attention back to our notes. So we defined stem cells, we defined differentiation, and then in that video, Hank talked about 2 different kinds of cells. So now we're going to get into what are our different kinds of stem cells. So again, in your notes, again, maybe skip a little space, maybe write a little bit bigger, maybe box it in. Again, giving your notes a sort of organization, an outline, a pattern helps organize the information in your brain. All right, so yeah, so like literally indent here, right, number 1, underline the word totipotent. So the word is totipotent, the root word toti meaning total. These are the most useful cells. These are the cells that can develop into any kind of cell, including the cells that make up the placenta and the supporting tissues for an embryo. These cells are exceptionally hard to get at because you can only get them when the cell is that blastocyst, before the egg actually implants itself in the wall and initiates a pregnancy. It's the cells that become, like I said, they can become any other kind of cell. So totipotent, total. And just so you know, they're listed in their order of their usefulness. So the most useful are the totipotent. Now, these cells aren't super important to scientists because, frankly, they don't really they're not really concerned about the cells that can develop into the supporting tissues, the placenta and the supporting tissues. So when we talk about embryonic stem cells, we're not really talking about totipotent stem cells. We're actually talking about these ones here, the pluripotent stem cells. So pluri meaning plural or many. So these are the cells that can develop into almost any other kind of cell. These are the cells that actually come from that inner cell mass. If you remember, I paused that video. Hank explained what, or Hank just briefly explained what a embryo is, or a blastocyst rather. So it's that sphere of cells. Those cells on the outside develop into the supporting tissues, the placenta, things like that. It's that cells on the inside, the inner cell mass. Those are the cells, when we speak of stem cells, when we speak of embryonic stem cells, those are the cells that the scientists use most frequently. And they can become almost any other kind of cell. The only cells they can't become the supporting tissues. They can't become the placenta and the supporting tissues in a pregnancy. So these are the cells that the scientists are really after, the embryonic stem cells from that inner cell mass. All right, so again, if you need to pause to finish getting down these two definitions, go ahead and do so. I'm going to get rid of this, and then we're going to move back to our next video. This one's only about 2 minutes long, and this one actually just takes us through what human development looks like. For whatever reason, the website doesn't let me full screen these, so we're just watching it like this.
In this video, then, we'll look at the beginning of development, looking into the ovary at an unfertilized egg. It then gets fertilized by sperm, which travel down the fallopian tube. So here, millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus. And then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring. And this is one of the early growth phases. As the embryo moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. Of course, in real life, that takes days, about 5 days. At this stage, then, I'd like to draw your attention to the inside of the blastocyst, where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal. And the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now we'll see those blue inner cell mass cells form a disc, and then as the cells continue to grow, they change their physical positions, their kind of geographical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo. Those lines represent sites where cells are migrating in and out. And here's an important stage when the three beginning layers of the embryo, the so-called germ layers, are formed, and I'll come back to that in a few minutes. As development proceeds, there's more growth and movement of cells. It'll begin to form a neural tube. Here it turns, and appendages start to bud out. You see the head forming and the eye. And then eventually we get a small embryo, and some months later, of course, this would be born as a young baby.
Okay, so just our first clip, just a quick whirlwind tour of human development. All right, so here's our second one. This one's just about 1.5 minutes long.
Here we're going to go through quickly the early stages of development again. And then we're going to get to this stage when the germ layers are set aside. The three germ layers, the ectoderm, mesoderm, and endoderm. So here you'll see then that the inner cell mass gives rise to the entire adult animal, in this case a human. This same process would occur in a mouse, as we'll see a bit later. If we now look at the embryo after the three germ layers have formed, this video will highlight what comes from the blue germ layer, the ectoderm.
Okay, so I just want to pause here for a second. So during development, this is really our first stage of specialization or of cell differentiation. So now those cells migrated, they form this little flat disc. And then here we have three layers. We have the ectoderm, the mesoderm, and the endoderm. You don't need to know what those terms are. But here, so these cells will give rise to the skin and nerves. And these cells will give rise to muscle and bones. And these cells will give rise to the gut tube. And the point is that this is our first level of specialization or differentiation. We now have cells that these cells will divide into ectoderm cells, not mesoderm or endoderm cells. These cells here will divide into endoderm cells, not mesoderm or ectoderm.
You'll see that it makes the nervous system, including the brain and the skin. The middle part, the mesoderm, green here, gives rise to the muscle, including the kidneys, the heart, and the endoderm gives rise to the whole gut tube. There you see the lung, the liver, the intestine. And to give one more detailed example of this, let's think about the development of the endoderm, and in this case, the formation of the pancreas. So there's the pancreatic bud, which comes out of the endodermal derivative.
Okay, so now pause it there for just a second. Here's another level of differentiation. Right, so here we are several weeks or months into development. So from the mesoderm, we now have, the endoderm, we have these cells, which is the pancreatic bud. So now we've gone from just completely undifferentiated cells to differentiated to endoderm, and then now those cells have specialized into the pancreatic bud. All of these cells will now divide only into cells that will develop into the pancreas.
Now I'm going to talk to you a bit today about the pancreas as one example.
All right, so now let's move on to our next clip. So I just got some notes here, make sure I stay on track.
The inner cell mast cells, which are going to be removed and grown in a petri dish. We first remove the outer layer that would normally form the placenta. Here the cells are put into a petri dish. Usually there are mouse embryonic fibroblasts on the bottom of it to help the cells grow. Most of them don't survive, but a few do and can grow into colonies of cells, as you'll see here. So this then is that process of self-renewal. The cells can continually self-renew, and in fact they are immortal in culture. They can be grown like this forever. Some mouse embryonic stem cells growing now in the lab were derived more than 30 years ago, so they're long outlived the life of the animal from which they came. Now here we're going to see them begin to differentiate, begin to start to specialize. So some of them are becoming mesoderm and some of them ectoderm. There you see a neuron forming. Here you see a different kind of tissue forming, and I'll show you an example of that in a second. One of the big puzzles in biology is to try to control this process now. So these cells with all of this potential that can become any part, how do we tell it what to do? Well, a growth factor, which has the funny name Sonic Hedgehog, named after the cartoon character, is actually quite important for telling cells what to do, as is another kind of a growth factor called activin. So these are examples of experiments where we're adding growth factors to cells to tell them what to do. And this is an area where we need lots of help from people like you to begin to figure out what.
All right, so I want to pause there for just a second. So yesterday we just had you read a couple of pages on cell cycle regulation, which is really just about cancer and getting our head around the idea that ultimately cancer is just the cell cycle gone haywire, cells dividing when they're not supposed to. We had you skip some of the pages on the regulation of the cell cycle, which is just crazy complicated biology. What goes on that causes a cell to divide, when it divides, how often, and during the life of the cell. So one of the more complicated elements in biology is really controlling this process of cell division. And there are so many steps involved, which is why cancer is such a difficult thing to solve, because there are so many different steps along the way where the cycle can get screwed up and get broken. which is why this is cutting edge research. It's just an example of how we still have so much to learn and so much to go. All right, last clip, and then we'll finish up the notes.
We're going to have a look at the way in which an amputated limb grows over the course of about 90 days in a salamander. That's a time-lapse movie, watching that thing grow. And now we're going to see what's really going on. Here's the salamander, and it's got a completely new limb. It looks perfect. It has inside bone. It has nerves and muscle, and it can even wiggle. In fact, it's perfect. Now we're going to cut it off.
Ew.
But here's the good news. It grows back. So don't be too scared. First, wound healing. Did you see that wound heal? Now we're watching what the newt can do that we cannot do. Cells are streaming out of the surrounding tissues into the area of the wound and forming what we call a blastema, which is a group of undifferentiated cells that are in fact really just like stem cells. And they're multicolored because they come from skin, from muscle, and even from cartilage. And these have a miraculous memory of what they used to be and are able to form a perfectly functional limb.
Pretty cool, huh? Something that we can't do. Actually, the one example of mammalian appendage regeneration is deer and the regeneration of the antlers that grow every fall and then fall off in the winter. All right, so let's turn our attention to finish up our notes. Okay, so we left off with defining these two kinds of cells, totipotent and pluripotent. We're almost there. So there's one more kind of stem cell, and it's referred to as multipotent. Let me get myself out of the way here. So a multipotent stem cell. So these are cells that can develop into a limited number of other kinds of cells. These are obtained from adults, and they're very difficult to get at because they are buried in tissue. So, for instance, if I damage, a bone, if I break a bone and my bone gets set by the doctors and then it starts to heal, it's the multipotent cells found in my bone that can develop into the different kinds of bone tissue. But multipotent cells from bone can't develop into, say, liver or skin or muscle. So these are three types of cells in their order of usefulness: totipotent, pluripotent, multipotent. These ones really here are the important ones, the pluripotent ones that come from that inner cell mass, from an embryo. These are the ones that are used in the research. All right, so what are some potential uses of stem cells? This is our last slide. All right, so potential uses. Stem cells could be used in actually a variety of ways. One, to grow new cells to replace damaged organs or tissues. Take, for instance, nerve cells. You're born with all the nerve cells that you have. And for whatever reason, that's not really all that well understood. Nerve cells are stuck in interphase. Nerve cells do not grow and divide once you have all your nerve cells, which is why paralysis, like if you break your spinal cord, paralysis is almost always permanent because those nerve cells don't grow back. Well, maybe someday we could figure out how it is that we could get nerve cells to grow back, and maybe paralysis would be a thing of the past. It's potential to grow new organs in a lab from stem cells. Imagine if owner-donor lists were things of the past. If you needed a new liver or a new kidney, you wouldn't have to wait for someone to die in a tragic car accident and harvest their organs. We could just go to a lab and we could grow a liver or a kidney right in the lab to replace the one that you need. Obviously, research on there's all kinds of different genetics diseases that we have a lot to learn about yet. And then cancer. If we can figure out how to turn off and turn on the cell cycle in stem cells, then maybe we can do that in cells that are cancerous inside a human body. They can be used to test different drugs for effectiveness. And then number 6 is important. Question mark. Who knows? There may be potential uses for stem cells that we are not even aware of yet. Stuff that is around the corner in the future, yet to be. All right, so that's it for the lecture on stem cells. Again, the big takeaway, know what a stem cell is, right? Know what stem cells are, know what the process of differentiation is, know the usefulness of these three kinds of cells, totipotent, pluripotent, multipotent. Be aware of some of the uses of stem cells. As always, supplement your understanding with the reading of 10.4. If you have any questions, don't hesitate to email or call. Excellent. Have a good day.